Assalamualaikum. So today I will be presenting about the molecular basis of gene therapy and stem cell therapy. So this presentation is divided into two. The first part will see the molecular basis of gene therapy, and the second part will see the molecular basis of stem cell therapy. The first part of the lecture, we will look at the gene therapy and how it works. First, we have to know the types of gene therapy. There are two types of gene therapy, which are classified according to the targeted cells of the therapy. The first type is gene therapy by using germline cells. Basically, in this type, gene therapy is done on germline cells, which are the sperm and ovum, which we know is the function is to deliver or pass the genetic materials from parents to the offspring. Therefore, when changes of DNA is made in germline cells, in these cells, um, it will allow for the permanent changes of any disease-causing genes that are certain to be passed down to the next generation. Also, we have to note that if this method is done early in the embryologic development, gene transfer can also occur in all cells of the developing embryo because, as we all know, the germline cells are the reproductive cells. The second type of gene therapy is gene therapy by using somatic cells, which are the non-reproductive cells. Um, in this type, uh, the targeted cells are the affected cells, which causes the disease. Therefore, it will not be passed down to the future generation. Therefore, this type is said to be safer and more conservative. However, the downside of this type is that it is short-lived because the cells will die and will be replaced with new ones. Therefore, repeated treatments is required to maintain the therapeutic effects. Now we will look further into the somatic gene therapy, which has two categories, ex vivo and in vivo. In ex vivo gene therapy, it involves modification of cells outside the body, which are then transplanted back in again into the body of the patient. The mechanisms are as follows. Uh, the cells from patient's blood or bone marrow are removed and they are let to grow in the laboratory. The cells are then exposed to the virus that is carrying the desired genes. The virus that is inserted or exposed are cells that elicits only weak immune response and does not cause any disease to human. And then when the virus enter the cell, it will insert the desired gene that it carries into the cell's DNA. The cells are then let to grow and then are returned back into the patient's body intravenously. The second category is in vivo. It is principally the same with ex vivo, except that it the gene transfer occurs while the cells are still in the body. This image shows the methods of somatic gene therapy. In ex vivo, the stem cells or the progenitor cells are extracted from the patient's body and it is let to grow in the laboratory, and then the virus which carry the desired gene are inserted into the cells, and then the cells are let to grow and then they are inserted back into the patient's body. In in vivo genetic therapy, the virus or the non-virus delivery vehicle which carry the desired gene are injected into the patient's body, and then the virus or the non-virus particle will go to the targeted genes associated with the disease. So we have looked at the types of targeted cells in gene therapy, and also the methods used to insert the desired gene into the targeted cells. Now we will look at the mechanism in which how the insertion of the desired gene causes changes in the cells. So there are three techniques of gene therapy, which are gene augmentation therapy, gene inhibition therapy, and also killing of specific cells. In the first technique, which is gene augmentation therapy, the aim is to treat diseases caused by a mutation that stops a gene from producing a functioning product. So basically, in this technique, the cell which have the non-functional gene will be inserted with a functional gene, and this will cause the production of product which will help the cells to function normally. In the second technique, which is gene inhibition therapy, basically the cell which contain the gene which causes the disease will be introduced by a gene which this gene will block the faulty gene, and thus the cell will function normally. The third technique is killing of specific cells. There are two ways in which this can happen. The first one is in this picture you can see insertion of suicide gene into the disease cells, and then the suicide genes will produces toxic product which will kill the disease cells. The second way is introduction of a marker gene, and then these marker genes will cause expression of marker proteins which will be expressed on the cell surface, and then these marker proteins will be detected or recognized by the immune cells in the patient's body, and then the immune cells will attack the targeted cells, and then subsequently cause death of the cells. This technique, the third technique, is suitable for diseases such as cancer that can be treated by destroying certain groups of cells.
Hello everyone, we are now on the second part of the lecture, which is stem cell therapy. I'm sure most of us have heard of this technology before, so this part of the lecture will comprise of three main points. Firstly, the mechanism of stem cell therapy, followed by the diseases that can be treated by this technology, and lastly, the applications of stem cell therapy. Before we go on deeper into the topic, it is very crucial for us to understand the three types of stem cells, namely embryonic, adult, and induced pluripotent stem cells. As you can see, the slide seems a bit wordy, but it's just an explanation of the three stem cells I've mentioned previously. In short, embryonic stem cells are the starter cells of our body, meaning they are undifferentiated and immature. And the most interesting part is that they can grow and specialize into any other kind of cell in our body. Next, adult stem cells. They are more mature than the embryonic ones, and they have limited ability to differentiate. They are then further subdivided into hematopoietic, mesenchymal, and fetal stem cells, which I'm sure we're all familiar with. And lastly, induced pluripotent stem cells. They are actually adult cells, but they are taken from an individual and then reprogrammed in a lab to become like embryonic stem cells. Hence, they have the capability to function just like an embryonic stem cell. Alright, let us move on to the mechanism of stem cell therapy. So how does it work? To make it simple, we will learn from this diagram of 6 stages of stem cell therapy. First and foremost, we will use chemotherapy and synthetic growth factor to move these stem cells from the bone marrow to the bloodstream. And once they are in the blood, a machine will collect the blood and separates out the stem cells. Next, the stem cells collected are then frozen and ready to be returned to the body later. Meanwhile, more and more chemotherapy is used on the individual to wipe out the bone marrow and immune system partially. This is to ensure the body will not reject the stem cells later. Then, to return the stem cells into the body, a drip will be used. Lastly, the body is led to recover for about 3 to 6 months, or sometimes longer. After understanding the basic mechanism of this therapy, let's find out the diseases that can be treated by using this technology. These include blood cancers, solid tumors, non-malignant blood disorders, immune and metabolic disorders. And in this table, we have listed some examples for you to look at. And as you can see, there are a lot of diseases that can be treated by stem cell therapy. Yay! We are finally on to the last slide of this subtopic, which is on the applications of stem cell therapy. Stem cells can be used in many ways, whether as a research tool in laboratories, or as cell therapies, as drug target validation, drug delivery, and many more. Have you heard of 3D printing of organs? For your information, stem cells are also used in this technology. The stem cells are integrated into the 3D printing inks, and voila, you get a 3D printed organ. Interesting, right? So it's very clear that stem cell therapy carries a whole lot of benefits, and I hope we can all benefit from it someday. Thank you.